So we'll go ahead. It's right on time to the moment here at 3 o'clock. I want to welcome you all. We're expecting several hundred people, and we're well on the way to that. Thank you. Uh, there are wonderful things about Zoom. There are headaches about Zoom. We're going to focus on the wonderful, and it does allow us a chance to get together without all the hassles. So first of all, please keep yourself muted, and we've got a whole team making sure that we're tracking our audio well, and we will be recording this. I think you all know that. And feel free to write questions into chat. We'll do our best to answer those questions in real time or during the small group discussions and such. Let's see here. So I'm going to give you just a couple highlights of what we're doing today, and then we'll have 5 brief topics about progress that we think are very exciting in Merkel cell carcinoma and wanted to share with you. There'll be a few minutes for some question and answers, and then some patient stories. We have 3 folks who have told their stories in a few minutes each. They're very different from each other, and we hope you enjoy those. And then while we take a break for 5 minutes, we will be moving into small groups, and we'll be assigning people. There's 5 topics for small groups. You probably know this. Blood tests, clinical trials, caregivers for patients, therapeutic vaccines, or newly diagnosed patients. So you can pick whichever one of those you want to be in, and they'll all be moderated and such. And you can hopefully in that setting have an easier time asking questions. I want to start off by thanking, look at all of these people here on this side. These are the folks that helped really organize the logistics of this and send out 232 little care packages. Maybe you got one kind of like this with your microwave popcorn and such, and a little ribbon that we made. So I hope you're enjoying those if you got one, and thanks to the team that assembled those items and helped us with all of the logistical planning here. In particular, Krista, who did a huge amount of the logistics and planning for this. We want to thank our donors. That has made what we can do much more feasible. Average donation is about $75, and over $7 million in total has been amazing over these past 15, 16 years with over 4,000 donations. Super impactful. How do we use philanthropy? Well, we train the next generations of physicians and scientists. You can see Geraldo and Samia here are our brand new PhD students joining Thomas, who's finishing up his MD PhD. And we have about 20, 25 people in the lab doing all different kinds of things at different phases of their careers. We are investigating promising new ideas, and we spend a little bit less time writing grants and a little bit more time focused on clinical and laboratory research that we think will really make a difference for patients. This is how we're using some of that philanthropy money. This was a very fun little thing to put together. Every person is next to a little pie chart indicating what fraction of their support comes from donations versus grants. You can see in aggregate, the donations are a big part of actually my ability to carry out this work. And you can see various folks are mostly grant supported or mostly donation, but across everybody, that's a lot of people being supported by donations, and we're very, very appreciative of that. I also like to thank our funding sources, the NIH, that has given over $20 million in competitive grants since 2006. Our research collaborators, many of you are on this call, and our industry partners, including these companies shown here that are some of our major long-term partners, but I've seen several other folks from our smaller partner groups where we're trying to get to trials going for the next few years. And then, of course, patients, your participation in research is the completely essential thing. Donating blood, donating tissue, donating information about how your health is going and which treatments you've had makes this all possible for us. And then we have many of our clinicians and clinical staff and our colleagues around the country. I see many of our close collaborators around the country on this Zoom event today. So in aggregate, it's pretty awesome, the progress that's happening. Here's my one slide on Merkel cell carcinoma. Most of this information and much more is available on our website or on other locations, but there's about 3,000 or a little bit more people every year in the United States newly diagnosed with Merkel cell carcinoma, and the number of such people is going up pretty quickly. The recurrence rate is around 40%, and it's a more aggressive cancer than melanoma, by several fold, more likely to come back. The risk factors, as you know, include older age, where we think the immune system is not as strong as we get over 50, 60, 70, and high sun exposure, fair skin, and immune suppression. As you know, 80% of Merkels are caused by this crazy virus that's on our normal skin. They're called the Merkel cell polyoma virus, and it very, very rarely causes this cancer. It doesn't cause any other cancers that we know of. And about 1 in 5 Merkels is caused only by sunlight. Treatment, early on, initial treatment is surgery and radiation. And when necessary, about in 1/3 of patients, when it goes farther in the body, we now use immunotherapy with 2 FDA approvals of Bavencio and Keytruda in 2017 and 2018 in studies that our team significantly was involved in. That has increased the chance of surviving advanced Merkel cell carcinoma by around 10-fold, which is amazing. But still, about half or more than half need extra help, and they don't have persistent benefit from these immunotherapies. So we have a lot of work to be done, and that's what we're talking about today. It's not a lot of sitting around on our laurels and congratulating. This is a focus on, we've made progress, we've got momentum, where are we going with the team? So we're going to organize today, here's kind of my last slide, focused on the patient experience, moving from diagnosis to initial treatment to surveillance where there's no evidence of cancer, but we're worried it might come back, to managing it if it does come back, and exactly how we treat that, especially if it doesn't respond to our initial treatments. So we're going to start off with Marika Birma, who's going to talk about our annual patient update project, which relates kind of to this surveillance period in particular, where we're looking to see what's associated with the cancer coming back or not coming back, and all of those risk factors and such. Then Dr. Lisa Zeba from Stanford will talk about blood tests. We've had one test based on antibodies for the past 10 years, and we have a new exciting blood test focused on circulating tumor DNA. And Dr. Zeba will tell you why that's a very exciting addition to our management. Dr. David Cole has for several years been leading our efforts to develop a therapeutic vaccine against Merkel cell carcinoma. And we have some of our colleagues who are involved in developing that on this call as well today. And David's going to give you a high-level view of how we hope to get that into patients starting in less than a year. That is the hope. And then Dr. Ganel, our current clinical research fellow, one of our research fellows, will talk about several clinical trials that are relevant for these various periods and various challenges. And we'll end with Dr. Bakuni, a postdoc in the lab, talking about a completely new area that we're very excited about that we think might help turn on an immune response in the patients who are not responding already to our current immunotherapies. And that's by doing something called Inhibiting Atr. So she'll be telling you what ATR is and why we are excited about that. So with that, I am going to let Marika take over here.
Thank you, Dr. Niam. Hi, my name is Marika Birma, and I'm an undergraduate research assistant in the Niam lab. I'm entering my 4th and final year as an undergrad at the University of Washington, where I'm seeking a dual degree in microbiology and the comparative history of ideas. And I eventually plan to attend medical school. Today, I'm going to be talking about our annual patient update project, which is more informally known as the PUPdate project. There we go. Sorry about that. So I'll be talking about a PUPdate project, and I've done PUPdate for 3 years now. At the high level, it's a summer project that the undergrads in the Niam lab do. We follow up with patients who haven't been seen in over 6 months. To do this, we double check medical records and patient information with that in our database to make sure that everything is accurate and up to date. This is one page of 14 pages in our database that captures relevant patient information. This PubDate project is extremely valuable because we have what we believe is the most comprehensive and detailed MCC database in the world. This database is ultimately the backbone of our entire research program. So whenever scientists or clinicians are looking to begin a research project, they often look to the data that we capture within this database. Therefore, it's extremely important that we're constantly double checking not only our own work, but the work of previous clinical fellows to make sure that this information is accurate. The PubDate project not only helps us improve data quality and maintain the database, but it allows us to respond to patients' questions and concerns. Part of PubDate is contacting patients via phone or email to get an update on their health, which is not only important to make sure that we are accurately following patients in our database, but also to farm off different questions to the relevant folks in our lab. This year, for PubDate 2021, we had 505 patients to review and contact. Our database is quite large with over 200 data fields that we have to double check every year. That's why it's great that we are doing this project annually so that we're able to make sure that all of that detailed information is accurately represented in our database. Our database is not only the driver of new clinical guidelines for MCC, but it also provides evidence for improvements in patient care. So it's important that we're constantly updating this information and double checking our work. Finally, I would like to acknowledge the other wonderful undergrads who worked on PubDate with me this summer. They include Alex, Emily Nguyen, Nikhil, Emily Gong, and Ali, shown here on the left at our undergrad lunch a couple of weeks ago, and here on the right, working hard on PubDate in our conference room. Next, we'll have Dr. Lisa Zaba to talk about MCC blood tests.
Hi, Lisa, we see you.
Great, and you can hear me too?
Yes, crystal clear. Welcome from Palo Alto.
Hello. All right, I think we've got this. Okay.
I don't see your screen yet, though.
Oh, you don't see my screen yet?
No, you might be on a different monitor or something.
Hang on, apologies.
Okay.
Let me try that one more time, and if not, I have another trick.
Okay.
Can you see?
I think we're going to see it. Yeah, it's popping up. Yep, just share full now. We got it.
You got it. Wonderful.
You got it.
Okay. Zoom issues aside, here we go. So thank you all for coming to this celebration, and thank you, Dr. Neem, for inviting me to speak. It's always truly such a pleasure to everybody on this call to speak to basically patients about this topic that we love so dearly. This is the entire reason that we do this work, is to help patients with Merkel cell carcinoma. So today I'm going to talk to you about blood tests that we are using to detect the early Merkel cell carcinoma recurrences. So why is it so important to accurately and quickly identify a Merkel cell carcinoma recurrence? Well, in Merkel cell carcinoma, as Dr. Neem already explained in his introductory slides, it has an extremely high recurrence rate of approximately 40%. This is compared to cancers like melanoma, which is around 7%. Secondly, and this should impress you, Merkel cell carcinoma grows extremely rapidly. Maybe some of you experienced this with your own Merkel cell carcinomas, but this is a photograph of a Merkel cell carcinoma on someone's left cheek that then over the course of the next 10 days grew about 6-fold into what you see in the right field. And thirdly, because of the high recurrence rate and the rapid growth, it is extremely important to identify these tumors quickly so that we have time to try a few different therapeutic approaches in the event that the first-line treatment is not effective. This is to do this before the patient gets sick and there's a large amount of disease burden. To use two common idioms, we need to catch things early to allow more shots on goal before the cat is out of the bag. So early detection of Merkel cell carcinoma falls into the large umbrella category of Merkel cell carcinoma surveillance. And the Merkel cell surveillance toolkit is multifaceted, each with very different uses. To the left here, you can see that I've discussed the full skin and lymph node exam. Are you guys hearing some feedback? Okay, hopefully that has ended. Okay, good. The simplest tool in the surveillance toolkit is the physical exam, and this can detect local and nodal recurrences large enough to see or feel. Relative to the other tools, this is a fairly blunt instrument, and so I analogize this to the hammer. Imaging with CT or PET CT is much more sensitive, and it can detect tumors that are greater than around 1 centimeter. And now we have 2 ultrasensitive blood tests that can determine if Merkel cell carcinoma is present in the body. The AMERC, which many of you are familiar with, is the polyomavirus serology test, and ctDNA is a new test that measures tumor DNA in the blood. AMERC has been around since 2009. This is the test that actually Dr. Neum helped to create with his lab. And ctDNA, we just started doing this in around 2020. I like to think of these as the X-Acto knives of Merkel cell carcinoma surveillance. So what is it about the AMERC that makes us need to seek out other blood testing opportunities? There are 2 main drawbacks to the AMERC. 1, 50% of patients do not have polyomavirus antibodies. This right off the bat excludes approximately half of our patients from this method of blood surveillance. So we need something else. Secondly, because the test uses the immune response amplification as the Merkel cell carcinoma detection method, MCC-specific immune stimulating events, such as a recurrence of the Merkel cell carcinoma, or the addition of immune stimulating agents, such as an immune checkpoint inhibitor, can cause this test to become unreliable. To demonstrate an example of AMERC unreliability, I'll take you through an AMERC serology timeline for a patient of mine. This is a 62-year-old male who initially presented with a 3-centimeter tumor on his right forearm. He had a positive sentinel lymph node biopsy. We excised the tumor and radiated the right forearm and lymph nodes. However, when he returned to his 3-month follow-up, an AMERC was checked and it was elevated, a significantly large rise in his titer. The PET CT was done very shortly after that, and it was positive for metastasis to the bones, and avelumab was started. The AMERC accurately diagnosed his Merkel cell carcinoma recurrence as it was intended to. However, just for learning and for me to teach the residents, I kept checking the AMERC over time so that I could have an example such as this. You can see as we do the PET CT scans, the patient is having less and less disease. However, at month 16, when I rechecked the AMERC, it had risen. This was a false positive rise in the AMERC. This simply demonstrates that the immune system is being stimulated by the immune checkpoint inhibitor, not that the patient is having increased burden of their Merkel cell carcinoma disease. And that was definitely demonstrated by the PET scan showing less and less disease. So given these limitations of AMERC serology testing, we began to explore the possibility of using ctDNA technology to detect Merkel cell carcinoma recurrence in all patients, including those who are polyomavirus negative. Circulating tumor DNA is cell-free tumor DNA that is found in the circulating blood that is shed from the tumor. In order to perform this test, you need a small piece of the patient's Merkel cell carcinoma tumor from their original biopsy that is then genomically sequenced to look for tumor-specific mutations. The ctDNA then looks for those patient-specific mutations in the blood over time in order to determine if there is a Merkel cell carcinoma tumor existing in the body at that moment. Because ctDNA in the blood degrades within hours, this test is an extremely accurate measure in real time as to whether tumor is present in the body at the moment the blood is drawn. So in March of 2020, we contacted the ctDNA company Natera to see if they would partner with us to develop a ctDNA test for Merkel cell carcinoma. They had already developed surveillance ctDNA testing in other cancers very well for breast, bladder, and colon cancer. They agreed to work with us, and they have been providing our patients with free ctDNA testing while we pilot the use of this new technology for Merkel cell carcinoma surveillance. The smallest primary tumor that we have collected to date is 0.6 centimeters, and we did see a ctDNA signal in that patient's blood. This means that ctDNA in Merkel cell carcinoma is more sensitive potentially than even a PET scan at detecting minimal residual disease. We also noted as part of our research, this is a logarithmic scale measuring the mean tumor molecules per mil in a patient's blood. And the x-axis is demonstrating the Merkel cell size when we drew that blood value. And we are demonstrating a logarithmic correlation with Merkel cell carcinoma size and the number of mean tumor molecules that we see in the blood. Therefore, not only might this actually be a great measure to determine if there is any Merkel cell carcinoma in the body, it may actually be a volumetric measurement. Further studies to come on that. This is an example of a patient whose ctDNA testing improved her patient care by allowing us to detect her Merkel cell carcinoma sooner. She is a 61-year-old woman who developed a rapidly growing Merkel cell carcinoma of the right cheek. She did not have any regional or distant mets when she was seen initially. Her AMERC was negative. However, we checked a baseline ctDNA, which was indeed positive, at a mean tumor molecules per ml of 32. Because of the large primary tumor size, surgery was held and she was started on 66 gray conventional radiation therapy. After the completion of her radiation therapy, she didn't have any apparent residual disease and her ctDNA had gone down to 0. This was at 3 months' time. However, she then had 2 subsequently rising consecutive ctDNA tests, which prompted us restaging her with a PET-CT. The PET-CT showed a hypermetabolic uptake in the right lateral thigh, and this lesion was biopsied and confirmed as metastatic Merkel cell carcinoma. Since her initial restaging scan was scheduled at 3 months, the ctDNA actually helped us to detect this metastatic lesion at approximately 1 month earlier. The patient was started on immune checkpoint inhibitor, and we continued to check her ctDNA. So sort of to sum this all up for you in a simple diagram, I'd like to quickly go over with you the advantages and the disadvantages of each of these tests. We tend to order them both at the same time because each of them have things that they bring to the table. So AMERC serology, again, as we've discussed, this can only be used in approximately 50% of patients, whereas ctDNA can be used regardless of the polyomavirus status. The AMERC serology, thanks to Dr. Niam's group, is an easily ordered test that requires only a requisition and can be done by any clinician across the United States. Currently, ctDNA is still under investigation. We're doing this across 7 academic centers across the United States. However, we do anticipate that once we fully vet this test, that it will be available widely across the United States. With AMERC serology, you do need a baseline test within 3 months of the initial treatment. And that is because over time, the AMERC serology falls to 0. And once you have left the 3-month window, you're not sure if the patient is either AMERC negative or if they simply had their titers fall over time. Whereas the ctDNA can be performed at any point during the patient's treatment or surveillance course and still be a good metric. The AMERC serology obviously provides information on the polyomavirus status, and the ctDNA does not. This is important because AMERC negative patients do have a 40% higher risk of recurrence compared to the patients where their tumor is mediated by polyomavirus. And then lastly, as we've already discussed, the AMERC serology is much less reliable after the first Merkel cell recurrence or during treatment with an immune checkpoint inhibitor, or like the next speaker is going to talk about, would also be less reliable during a therapeutic Merkel cell carcinoma vaccine trial. The ctDNA is not dependent on the immune system to be a readout, and therefore can be used during any of these immune modulating events accurately and adeptly. With that, I conclude and give the thanks to, why is my slide not turning? There we go. And I give great thanks to my wonderful mentor, Dr. Niam, who's totally fantastic, as you're all aware. And then to both his team and my team that have been instrumental in carrying forth the AMERC test as well as ctDNA testing. Without further ado, I know we're on a tight timeline. I'm going to introduce Dr. David Cole, who will speak on the therapeutic Merkel cell carcinoma vaccines. I'm going to stop sharing.
Thank you very much, Lisa. And I'm getting a thumbs up from Paul. So I'll
yes, David, you're crystal clear. And
launch right in.
Looks like sharing is working great. Yeah, thanks, Lisa.
Yeah.
Big
And,
progress.
Hello, everybody. Good afternoon. Some of you I've met before, and it's a pleasure to see so many folks on my other monitor. And it's wonderful to have a chance to give you a brief introduction. I'm not going to be as technical or as many slides as Lisa's were. This is going to be a little bit higher level about our concept of a potential therapeutic vaccine to prevent Merkel cell carcinoma from recurring. And I'll start by saying we're targeting that approximately 80% or so of Merkel cell carcinomas that do have the virus in them, the Merkel cell polyoma virus, mostly with this work. So as looking at the timeline at the bottom where Paul helped us sort of map out this afternoon's program, the spot that we're looking at kind of in the, for with regards to patient selection, at least initially, and in sort of the timeline, would be in those individuals who have a local Merkel cell carcinoma, something that can be treated, and showing with the red arrow here, either with surgical removal or sometimes combined with radiation. And then somebody who's had a staging and has no known metastatic or widespread disease. And then we're in kind of a waiting game. And the standard of therapy at this point is surveillance, that Lisa just spoke about. But what we know is that these such individuals, and some of you may have been in this boat, are at quite a risk for recurrence. And the number is 30–40% over the first couple of years, something like that, but enough to be sort of frighteningly high. And what we don't have at this point is a FDA approved or endorsed sort of adjuvant therapy, adjuvant meaning helper, but something to try to reduce that risk of recurrence in people who have had their initial tumor dealt with and then are nervously, sort of waiting. So that's the patient group that we're going to try to address, at least initially. And let me see if I can successfully go on to next slide. So preventative vaccine given to block the initial infection or disease. This is not what we're talking about. We're talking about instead this concept of a therapeutic vaccine, which we would give to people who have had the cancer. The cancer's been removed. And you might say, okay, this is kind of crazy. Do we have any examples of this? We have one licensed therapeutic vaccine, which some of you may have had if you're like me, over 50 considerably. Just got my second one the other day. And that's this Shingles Vaccine, which we give to people to prevent shingles. That's an infection that people have had ever since they were children. They had chickenpox, and we're preventing that virus from causing a problem. So we have a little bit of a precedent for a therapeutic vaccine to boost an immune response. And this is a very active area of investigation in cancer to try to bolster that inherent immune response that people have that's trying to kick the cancer, it's trying to kick the virus that causes the cancer, but needs a little bit of a help from the vaccination. So how would this work? What's sort of the concept here? And Paul's team is better at graphic arts than I am, so they made this nice graph over on the right. As Lisa just introduced, when somebody has surgery that's maybe combined with radiation to remove the initial tumor, the y-axis here you can just think of as the immune response. Here we're focusing on T cells, this particular part of the immune response. This could be antibodies also. But with time, then when the cancer is at least initially gone, the immune response goes down. And what we're worried about is if the immune response goes down, that eventually if there's a residual tumor cells, just a micro locus, so just a little bit of tumor, that the virus may be able to come back. And the tumor may be able to come back. So we'd like to give the vaccine at some time point after the initial therapy to bolster that immune response with the goals of maintaining and rebuilding a robust immune response against the tumor virus in this case, and with the idea that these T cells, this part of the immune response targeted against Merkel cell, can eliminate any microtumors or any small foci of cells that may be sort of hovering, semi-dormant, but waiting to come back to cause a problem. So to pursue this goal, we've been lucky enough to partner with 3 different groups of people, and we have 3 platforms that we're actively exploring. I'm going to start by showing you the most advanced platform, and I think we have some individuals on the call from the firm. So a little bit of data, but first a few introductions. So Dr. Claire Rosian, who has done the animal experiments I'm going to show you just briefly, and the leadership of this company, Dr. Terry Hyland, who is really the originator of this particular vaccine concept. And this vaccine concept is the format, if you will. It's a so-called DNA vaccine. We have some time later in a breakout group, and I can talk about the styles of the vaccines and the format. We all know about, mRNA vaccines and everything for COVID. So I'm not going to go into that really much at this point. But just I'm so pleased to say that our biotechnology partner, Immunomics, has really committed to showing some efficacy in an animal model, and also manufacturing a pure batch of the vaccine for clinical trials, which is not a trivial pursuit, and also doing the required toxicity testing required by the FDA, which is also not a trivial matter, and actively engaging in a lot of work to actually get this into humans in 2022. So a tremendous amount of administrative and medical and legal work to get this actually going as a clinical trial. The data that I'm showing you here on the bottom are from a mouse model of Merkel cell carcinoma. There are complexities. We could talk about this in a small group of people who are interested. But just to sort of pay attention to the tumor size here is on the y-axis on the up and down, and the black animals were untreated. And as you can see, there was a great delay in this system where the tumors were extremely delayed in animals treated with the candidate vaccine. And this is summarized sort of over on the right in terms of the percentage of animals that were tumor-free. So I don't want people to get too either positive or negative looking at these red and black lines. This is quite an artificial animal system. The punchline is it showed activity. There was a strong immune response. I'm not showing you the immune response here. And there was also some real life tumor delaying, tumor shrinking activity. So so Immunomics has committed to moving this forward to clinical trials, which is great. And in my last slide, I just want to briefly review two other vaccine concepts that are less advanced, but which we're really excited about. The first one I'm just calling biotechnology partner number 2. The company name is over on the right if people are interested. And these people have a kind of a unique vaccine platform, one that actually hasn't gone into humans very widely yet. But they've also committed to producing vaccine for test tube and animal testing. And we're pleased at the University of Washington to be pursuing both the test tube, which looks good, and the animal model testing for their vaccine concept. And then thirdly, Dr. Niem was the recipient of an American Cancer Society grant that I've been helping him with. And in partnership with a third biotechnology company identified down here on the lower right, HDT Bio, we've been pursuing an RNA vaccine. So some of you may have received a COVID shot that was an mRNA vaccine. This is a variant of the mRNA vaccine technology. So it's similar to the FDA licensed product. And similarly, their candidate vaccine, the prototype, which we've been doing test tube tests on, it looks good. And these parties will be supporting us doing some animal testing there. So these, we're trying to get a couple of shots on goal here. We're trying to keep an open mind about which one or ones might be the most potent. Maybe we'll need to combine with the overall goal of bolstering the immune response in individuals who are in, for now, in that niche but large category of the initial tumor has been removed, and now we're in that sort of nervous, watchful waiting timeframe. So with that, I'd like to switch gears to Dr. Ganel, who will be actually moving forward in the clinical trials world to actually trials which are now currently enrolling, and will teach us about some clinical trials that are currently underway. And let me see if I can stop sharing.
Thank you, David.
Thank you so much.
We're excited to explore that space David's talking about. There is absolutely nothing available for patients who really need something to help lower their chance the cancer will come back. Take it away, Dr. Ganel.
Great, thank you. I'm so excited to be with you all here today, and I've had the pleasure of meeting some of you, and I would like to talk a little bit about some selected ongoing MCC clinical trials that we're doing. So as many of you know, the mainstay of cancer treatment for many different cancers, not just MCC, has been chemotherapy. And chemo is great because it kills cancer cells, does a really good job at that. But chemo is also really bad in some ways because it kills healthy cells, most importantly, the T cells of the immune system that are so important at keeping cancer at bay. And so what we see with patients treated with chemotherapy is although it can work quickly and work well, also quickly cancer tends to come back because the immune system is no longer working. And as we've heard already, we've had a lot of updates in cancer care, including MCC in the last 4 years, including the FDA approval of new immunotherapy, which is really exciting. And this unleashes the patient's own immune system to kill the cancer. And in most patients, it will keep healthy cells healthy. And so what this means for post-treatment protection is that people tend to not see their cancer come back as quickly or as readily after immunotherapy. So we have a few ongoing questions though. For example, does immunotherapy after the initial treatment for MCC prevent recurrence? So especially in patients who have more localized disease, they've had their initial surgery, they've had radiation, and their cancer, we can't detect it anymore. What about them? Because some patients in this category of surveillance, like Dr. Cole was talking about, can have up to 80% chance of their cancer coming back. And we're continuing to enroll in trials that ask the question, does immunotherapy in these patients prevent their cancer from coming back? Our ATOM trial uses avelumab with trade name Bavencio, and the STAMP trial uses pembrolizumab with trade name Keytruda. So I want to just orient you here to our timeline. And again, these trials are looking at patients who are in this surveillance period, this period of kind of nervous, watchful waiting. Is my cancer going to come back? And I want to compare the two trials for you really briefly. In the ATOM trial, this takes our patients who are at highest risk of cancer recurrence, stage 3 disease at diagnosis, comparing that to our STAMP trial, which takes in a wider range of initial stages at diagnosis from stage 1 to 3. The ATOM trial specifically uses immunotherapy and compares it to placebo for over 2 full years, whereas the STAMP trial compares immunotherapy just to observation for 1 year. But again, the most important similarity between these two trials is that patients cannot have evidence of disease when they enter the study because we're looking to see if these medications prevent their cancer from coming back. Well, that's great. But a lot of patients with MCC will have their cancer come back. And what do we do when cancer recurs or when cancer is widespread to begin with? Certainly immunotherapy has been our mainstay, but in a lot of patients, up to 40%, immunotherapy doesn't work or stops working. What do we do then? The first trial I want to introduce to you is being conducted by one of our research physicians, Dr. Joshua Veach, and it looks at taking T cells from a patient's body, removing those, adding a modified gene which helps those T cells to detect and kill MCC, and then putting the T cells back into the patient's body while the patient continues their current approved immunotherapy so that hopefully synergistically those two things will come together and kill their MCC. Another interesting idea, and these next 3 trials I'm going to talk about are being conducted by Dr. Bhatia, another one of our research physicians, also answering this question of what if immunotherapy stops working and how do we treat recurrence of MCC, is looking at a new immune stimulating drug that's directly injected into tumors that we can access either in the skin and soft tissues. What we think this does is stimulates the immune system right at that cancer area. And then with that, it teaches T cells, which then can circulate throughout the body. Our hypothesis is that then those T cells, which have learned what the MCC looks like and can go attack it in other places of metastatic disease in the body. And again, in this trial, patients are continued on their currently approved immunotherapy, so pembrolizumab or avelumab. We've seen some really exciting patient responses to this trial already. Recently, we had a patient whose tumor was injected, and she also had metastatic disease throughout her body. And within just a few months of initiation of this trial, all of her tumors had disappeared. So even in these trials, we don't have full data yet to share with you, but it's always exciting to see these initial responses. The final 2 trials I want to talk to you about in this area of treatment or of recurrence and what treatment we're going to use a kind of a different idea. So this idea I want to introduce to you is called a bispecific T-cell engager. Basically, this is a little molecule that you can think of as glue, which connects T-cells of the immune system to a target. So in this diagram here, you can see a T-cell, an all-important killer cell of the immune system. And these BiTE molecules here are there to attach that T-cell, in this case, to the cancer. This trial does this specifically with a receptor called somatostatin that's expressed by Mcc cells. And what's also really important about this trial and the next trial I'm going to talk to you about is that patients do not need to be on PD-1 immunotherapy. And this is really important because some patients don't tolerate PD-1 immunotherapy or can't use it or it hasn't worked. And so for those patients, these can be especially useful trials. The last trial in this same kind of idea, a different kind of BiTE, is actually connecting that T cell, not to the cancer itself, but to some bad acting immune cells that tend to let the cancer run rampant. So it's the same kind of molecule, but a different target. The trial that we're looking at here is called Ampavina, and the target that it's looking at is actually a little molecule that is expressed on these immune cells. And again, patients don't need to be on PD-1 immunotherapy, which can be a huge benefit for people that can't tolerate that. So last but not least, I just want to go over really quickly where we're exploring some of these trials. We know that most of you, as we heard at the beginning, are not near Seattle, and so it's helpful to kind of look at, hey, could I be involved, and is there a trial near me? The first two trials I want to talk about, ATOM and STAMP, were the two that you did not need to have active disease, and these were during the surveillance part of someone's MCC timeline. You can see ATOM has a few spots around the United States, but STAMP takes in almost the entire United States. So two slightly different trials and lots of options there. Next, the ATTACK trial is only here in Seattle. Our Execure trial does take in a little bit more of the country. This is the trial that has the injected medication. And then the last two trials where we were talking about new BiTE technology are in a few different spots around the United States. So I hope that's helpful to kind of see where there might be a trial near you. So I want to hand it off next to Dr. Balkany, and she's also going to talk about a really exciting new MCC trial.
Thank you, Dr. Ganil.
Hold on just a second.
Thank you so much, Lindsay. And Rashmi, welcome. We can hear you well, and we're starting to see your screen. And I'll just say, this topic, everybody, is really exciting and really tricky. But Dr. Balkany has worked very hard to get it hopefully clear to you, and we can hopefully share some of that excitement with you about something we're very optimistic about for the future. Thanks, Rashmi.
Sure. Thank you, Dr. Neem. Hi, everyone. Thank you so much for joining us. It's my pleasure today to be talking about can inhibiting ATR help MCC patients who do not benefit from immunotherapy? I'm going to tell you about an exciting clinical trial that is on the horizon. My name is Rashmi Bhakuni, and I'm working on this project in close collaboration with Dr. Pete Goff. Of course, you probably are not aware of what ATR is really about. And that's what my goal is for today, to tell you why we're excited to test this ATR inhibition in Merkel cell carcinoma patient who remain non-responsive to immunotherapy, which happens to be a case which happens to be happening in a fraction of MCC patients. To begin with, one key question that most of us are keen on knowing more about is, how do we treat MCC that has spread throughout the body, which happens to be the case in about a third of patients. And a part of it is answered by the use of immunotherapy. That has been a huge success in the treatment of Merkel cell carcinoma. So this pie chart here talks about the immunotherapy response in MCC patients with advanced disease who need systemic therapy. Our team was greatly involved in leading the clinical trials that led to the FDA approvals of Bavencio in 2017 and Keytruda in 2018 for Merkel cell carcinoma treatment. This has been helpful in providing benefit to approximately 50% of Merkel patients with advanced disease in an ongoing way. along with also providing up to 10-fold increase in the survival of these patients. However, the remaining 50%, as represented by the yellow and red sections of this pie chart, are able to show only a brief response or no response to such treatment. So, how do we help these folks in yellow and red that happens to represent a significant problem in MCC today, along with in a number of variety of cancers? One such promising approach that we are actively trying to learn more about is whether using an ATR inhibitor can help address this existing problem. So, what is ATR and why do cancer cells need it? Well, ATR, originally a protein with a long complicated name, in its most simple definition can be taken to be this checkpoint officer, that is Mr. ATR, that is responsible for verifying that a DNA is fully copied before a cell divides. By doing so, it provides a certain quality control mechanism within cells. What do I mean by that? Basically, a cell dividing is a complicated biological process required for growth, wherein one cell doubles itself over time by dividing into two. Normally, these cells are controlled by something called a growth checkpoint, which assures that these cells don't keep dividing when they shouldn't. However, these growth checkpoints are lost in case of cancer cells, because of which they keep dividing and keep growing in number aggressively. And this happens by these cells constantly splitting into two from one. Now, how do these two cells come from a single cell? This happens by copying of 6 billion pieces of DNA information per cell. And if this does not happen properly, the cell will go on to die. Now, coming back to the main point of how ATR works. So, Mr. ATR here acts as this quality control guy and attests to the fact that DNA here is fully copied before it allows the cell to divide. And since cancer cells are growing so quickly, they are in more need of this quality control mechanism. So, how is its inhibition useful? Let me explain this to you by taking the example of normal and cancer cells. As can be seen here, normal cells on the left are growing slowly because they have intact growth checkpoints. In contrast to that, cancer cells, as we can see here, are trying to break away from their site and increase in number by growing on top of each other. This happens because they have lost their growth checkpoint. Now, because cancer cells are in constant need to produce more copies of themselves, they have been studied to be more dependent on ATR, which has made this protein an interesting therapeutic target for more than a decade. Now, addition of an ATR inhibitor, that is an agent interfering with its function, will prove to be selectively more toxic for cancer cells, because these cells will then become irresponsible and keep dividing before actually having copied their DNA properly, and thus result in cancer cell killing. Interestingly enough, two major updates on the same front that have gotten us really excited include: one, availability of highly selective and potent ATR inhibitors for clinical use after almost a decade of delay, and two, ability of ATR inhibition to raise an immune alarm in a rather immunologically silent tumor environment by these dying cancer cells. So, how does inhibiting ATR activate an immune alarm? Well, here is a picture of some MCC cells that are rapidly growing and in a way cold in their behavior because they're not visible to our immune system. In the setting of an ATR inhibitor, and this is what Dr. Goff and I are starting to see in our lab now and remain quite excited about, these cells are made to call out to the immune system by starting to activate some specific signal that help attract some of these fancy gray colored immune cells. These cells in turn help generate a robust anti-tumor immune response by making these cells more visible to our immune system, along with inducing cancer cell killing as a direct effect of this inhibitor. The net effect of this can be summarized as turning an immunologically cold or silent tumor into an immunologically responsive or hot dying one via immune signal activation. Supporting this hypothesis is a recently published review article by our team, which describes in detail some high-level concepts of how this treatment will likely benefit non-responsive MCC patients. And it's now time to take this forward to a clinical trial and see if it really does work. As further evidence and support for this concept, some recent data presented at Big Cancer ASCO meeting this year showed some encouraging result in a related cancer, that is melanoma, of an ATR-based clinical trial, ATR inhibitor-based clinical trial. The subjects of this trial included melanoma patients who do not respond to immunotherapy, a similar significant problem that we face in MCC today. These patients can only be helped with drugs in a very small fraction of patients, that is around 25%. So this trial essentially tested the combination of an ATR inhibitor with immunotherapy and observed 60% benefit in those enrolled patients, wherein 30% of them had their tumor shrink, with an additional 30% attaining a stable disease state wherein the tumor did not grow. Overall, this combination was found to be well tolerated with very few side effects. These are some early data, but still encouraging for melanoma. And we are more than encouraged to test this same combination in Merkel now. So as we move forward towards a clinical trial, we have assembled some wonderful teams that will help us test this combination in Merkel cell carcinoma patient, with some early funding from the Mark Foundation supporting our work. So our team at Atr Pharmaceutical here has come up with a very promising ATR inhibitor that is ready to go into clinical testing phase, while our Cancer Immunotherapy Trials Network team has been instrumental in carrying out various trials that has provided significant benefit to a class of MCC patients. Lastly, we have been busy working closely with each one of our collaborators to help strategize the various aspects of this upcoming clinical trial aimed at exploring the combination of ATR inhibitor with immunotherapy, wherein our primary aims would include assessing the safety of this combination, along with understanding its potential to shrink tumor and provide long-term benefit. With that, we'd like to thank our entire team for always bringing in great ideas and providing a solid support system as we continue to work towards our shared aim of providing MCC care. Thank you.
Bravo, Rashmi. That is a really, really tricky topic, and I think you summarized it very clearly. Now, let me grab, we're going to move to Krista. Tell me if I'm right here, but I think we're on to some questions and answers now. Is that right?
Yeah, we have about 10 minutes, and Candace and I have jotted down a few of the questions that haven't been answered in the chat. It was actually very helpful for people to respond to some specific questions. We're going to focus on questions that don't have designated breakout rooms to begin with. And then if your question hasn't been answered, save that question for that breakout room. And if it still hasn't been answered, email us at mccteam@uw.edu. So I guess the first question that we identified was, should MCC patients get the COVID vaccine and booster? And also, should they get the flu shot this year? So
I can't think of a reason people shouldn't be getting those things. We have an infectious diseases expert on there. David, do you want to comment?
I agree completely, Paul. And I guess I'd say we've done some research in my lab, but people are concerned. They're saying, okay, I'm immune compromised because maybe I'm on some cancer chemotherapy now. Should I get the shot and so forth, or is the shot going to work for me? And we do have some guidance, I think. First, if you're older, and Merkel cell goes with being older, your COVID can be a lot worse. So definitely that's a reason you want to get vaccinated. If your immune system isn't so good because of maybe some chemotherapy that you're on, you also have a chance of having worse COVID-19. So that's another reason you want to get vaccinated. We have examples where people have gotten boosters and we've proved that they've had, and maybe they initially didn't have an immune response to the FDA licensed vaccine, and then they got a booster and then they did. And we now have some guidance from the federal government about boosters in such individuals. So I can't think of a medical reason other than allergy to one of the components of the vaccines, which is a serious thing and is rare but can occur, not to get vaccinated. The question about the influenza vaccine, we had a strange winter last winter with almost no influenza because of masks and social distancing. And people were worried about the rebound. What's going to happen maybe if immune responses in the community went down, and is it going to come back with a vengeance? We haven't seen that yet for flu, but for another infection called RSV, it's a respiratory spread infection, can affect kids, but also adults. We're seeing now with a vengeance, kind of this like, we waited and now we've got extra steam and we're coming back with kind of a vengeance to have a very strong RSV season. So I would definitely recommend also getting one of the quadrivalent flu vaccine products this fall.
Good. Thank you.
Thank you, Dr. Cole. I'm so glad we had you on.
Yeah.
Candace, do you want to ask the next question?
Yeah, so we had a question about kind of comparing X-knife or stereotactic radiation for MCC instead of single fraction radiation, and maybe the differences or different areas you might use those types of radiation.
And Candace, I didn't understand all of your words. You're comparing radiation that was single fraction versus what?
X-knife or stereotactic radiation.
Oh, gamma knife.
Gamma knife,
yeah,
yeah.
okay. Yeah, apples and oranges. I kind of feel like we're in a weed zone here, and maybe we should deal with that in a smaller group. But at the high level, we didn't talk about single fraction radiation, but it's a wonderful way to give one slightly bigger dose of radiation to clean up a possibly involved zone of Merkel after surgery. That is very different than gamma knife or stereotactic radiation, where you're zapping something in the middle of the body and you're trying to cure an obvious cancer. So we use them in pretty different ways, and I think we'll leave it at a high level answer at that.
Okay, so another question someone asked was how to monitor for recurrence risk after 10 years and what to look out for. And I think that maybe this individual might not have the Amr test, or probably not circulating tumor DNA test, because that wasn't available 10 years ago. But I'll let you answer that.
Yeah, another thing we didn't talk about today, which we're excited about, and I see Dan Hippie and a bunch of members of our team who helped develop it, is a recurrence risk calculator that any of our active patients are probably very familiar with. But it's really amazing how different risk is depending on whether you're immune suppressed or not, or what stage you're at, and what age you are, and whether you're a man or a woman. And this calculator can really plot that. And that's a very important tool. If your risk of having this cancer come back went down to less than 2% by 4 or 5 years or something, and then you followed it for another several years, and we've got some people who are maybe out for 10 years, and your cancer has never come back, we really think a lot of those people, and there was a little bit of that going on in the chat, don't need to be followed actively anymore. Once your risk is, so much lower than 2%, you don't need to do anything heroic anymore. It's different, you know, if there's immune suppression or there's a recurrence that's happened or something, but everything's customized and that's how it should be, personalized, and we can track that now with our recurrence risk calculator, and we're happy to work with folks, point them in the right direction so they can get that information, and that they don't do scans and blood tests that are unnecessary and expensive. We don't want that to happen.
And we can put that calculator in the chat so people can reference that. And we can also include it in a message to thank people. So you'll have that.
And I think there's a question that might be broadly interesting from Dr. Marcus, Robert Marcus. Do patients who also have chronic lymphocytic leukemia, CLL, and or B-cell dysfunction, which goes along with CLL, respond to standard immunotherapy protocols? And the answer, Robert, is yes, sometimes, but probably less than half as likely. And having a problem with your immune system for whatever reason lowers your chance of responding. Different things change it quite a bit. We think CLL cuts the chance of response to something like Keytruda or Bavencio in about half, or perhaps even more severely than that. But there's still hope, but it's a tougher battle for sure. And there's a question about how long should immunotherapy continue? That's an enormous question that we probably should have addressed more substantially, but one of the reasons we didn't is because we don't have definitive answers. The answer is probably more than 1 or 2 years, if at all possible. We are seeing the cancer come back in approximately half of patients after they stop immunotherapy, usually 6, 9, 12, 18 months after they stop immunotherapy. You're less likely to have it come back if you had a complete response to immunotherapy, maybe 40%, and you're a little more likely to have it come back if you only had a partial response to immunotherapy and you stop immunotherapy. Then it's more like about 60%, it looks like. We like, therefore, to not stop immunotherapy cold if possible, at least not until several years have gone by. And we think we can cut the dose or increase the frequency by maybe 4-fold and still have benefit, but go down in intensity a lot. And that's what we're doing with a lot of our patients and having good luck with that. But this is a very active area. It appears Merkel is a little more likely to respond to immunotherapy than melanoma, and a little more likely to recur after immunotherapy is stopped than melanoma. But we have a lot to learn in that area, and probably by next year, we can really hit on that topic in greater detail.